I'd like to give one final web lecture on my own collaborative research with colleagues at UC Davis on plant-associated microbes. This is to give you a feel for some of the things that we can now do with improved sequencing technology to characterize close species relationships. So no organism, no eukaryotic organism, animal, plant, or fungi, occurs without microbes nearby. We know that microbes associated with plants in particular do a number of things. They can provide defenses. In some cases, microbes such as endosymbiotic fungi create defensive compounds like toxic alkaloids. Some of the outbreaks of hysteria in the Middle Ages, some of the witch burnings, were in fact caused when ergo infected rye and wheat in Europe and the US, leading to communal madness and some witch burnings. Many microbes also provide important novel metabolism. In aphids and leafhoppers, internally housed microbes, such as Buchnera microbes, help these insects metabolize a diet that is essentially just sugar and convert these sugars into amino acids. In plants, associated microbes are important for helping plants get nitrogen out of the atmosphere from inert N2 and getting phosphorus, which is often immobile in soil where it gets adhered to soil particles. These microbes help digest it out of the soil. Another thing that microbes associated with plants and probably animals can do is manipulate their hormonal metabolism or growth patterns. So if microbes can create or make our own hormones, they can manipulate our growth by doing so. And it's been found that microbes associated with plants, bacteria or fungi, can make most plant hormones like abscisic acid or ethylene or auxin. And by making these, they likely have great effects on plant growth patterns. So I'm going to focus on nitrogen, which is a key limiting resource. So about 30% of our food, of the nitrogen in our diet, comes from legumes. Some of us may eat a large portion of legumes in our diet, but even without them, they're important as food for livestock or other animals because they're rich in protein, consequently nitrogen. And the reason that they're rich in nitrogen is that legumes associate with rhizobial bacteria. These are bacteria that can convert N2 gas in the atmosphere into ammonia and therefore into a building block of amino acids and proteins. And these rhizobial bacteria in plants are housed in a specialized organ called the nodule in the roots of plants. And in this nodule, they're kept in a low oxygen environment where they're able to do the magic, the conversion of inert N2 gas into ammonia in the absence of high amounts of energy. They do use sugar for this process, and that sugar comes from the plant host. Now, in crop legumes, the fixation efficiency, the amount of ammonia or amino acid generated from the rhizobia, tends to be lower than that of uncultivated legumes. And in many agricultural lands, think of the American Midwest, there's high soil nitrogen levels because of fertilization of other crops like corn or wheat. And many crop legumes have low levels of genetic diversity due to domestication bottlenecks and breeding. Now, we understand a fair amount about how rhizobia associate with legumes. There's a well-characterized pathway where the bacteria, like Mesorhizobium loti, create a set of flavones that are used to signal to the plant that they're willing to associate. And there are receptors on the surface of the plant that are key to this relationship as well. And if the plant exudes the right signal, its own flavonols, and the bacteria responds with a nodulation signal, the bacteria will enter the plant through a root hair if they have matching receptors. And much of what we know of this comes from studies where the gene of one partner, the plant or the bacteria, is mutated in a mutagenesis screen, and the consequences for the phenotype of one partner or the other is examined. But this tells us little about legumes in wild or agricultural settings, which have not had quite this attention in mutagenesis screens. So we still know a little bit about the diversity of each. So if we look at a phylogeny on the left of the plants and on the right of the rhizobia, we note that there are differences in specificity. So glycine, that's soybean, it associates with two distinct groups of rhizobia, Cynorhizobia and Bradyrhizobia. And these are distantly related groups of rhizobia based on this phylogeny on the right. The rhizobium that was first identified, Rhizobium legusarium, associates with many different legume hosts. It associates with medicago, plants in the same genus as alfalfa, pisum, which is pea, and fava bean, Vicia faba. And another, Mesorhizobium, associates with chickpea, which isn't even on this phylogeny because it's a minor crop, but is somewhat closely related to alfalfa. So there are differences in different legumes in which bacteria they'll associate with, and the same in the bacteria. And part of what may drive this is something called the pangenome. So I mentioned in class that a group had sequenced 3 different strains of E. coli. And these strains of E. coli differ by a full 40% of their genome. So only just about 40% of their genes are shared across all 3 of these E. coli strains. There are some genes that are unique to each E. coli strain, one of which is the standard lab strain, another of 2 of which cause different types of bacteria. So there are different sets of overlapping genes in these. And bacteria seem to be able to differ quite extensively in their genome, even though they still share some genes and share an evolutionary past. So when legumes colonize a new area, there's a reduction in population size. And in that new area, they may or may not have suitable rhizobial partners. And understanding this partnership can be important both for conserving legumes, say if they lose habitat to construction or habitat loss, and it can be significant in agriculture if we bring a legume to a new area where we might want to grow it. So we can visualize this population bottleneck with some alleles being lost from the wild as it's turned into a cultivar or an heirloom variety. And with modern breeding over the past century, there's a further erosion of genetic variation, although yield increases. So we have little sense of what the effects of these bottlenecks is going to be on microbial symbionts. And that's one of the things that my lab and our partners are trying to understand. So I'm talking about a project that was funded by the National Science Foundation through its BREAD program, that's basic research to enable agricultural development. And we worked with partners in India at an FAO-funded institute called ICRISAT, the International Crop Research Institute for the Semi-Arid Tropics. And this features some of the work of 2 of my graduate students, Vanessa Sanchez and Clara Scharnagel, and is really also a key part of the work of a UC Davis graduate student, Alex Greenspan. So we focused on India, where we grew chickpeas, featured here on the left with Alex and our partner Rajiv, and they're digging up the roots of chickpea. And on them we find these nodules, which we cleaned in the field to remove as many bacteria on the surface of the nodule as possible. So in total, from a planting of chickpea and wild chickpea in India, we collected about 2,500 nodules, and we extracted DNA from 1,800 of them. And out of these, we sequenced all of the DNA in the nodule, both plant and animal. We did it using a short read technology called Illumina sequencing, which I'm going to explain further in class. It means we sequenced all of the DNA in the nodule in 100 base pair pieces. So we have to put those pieces back together to figure out who is attached to who. So we generated a ton of DNA sequence data. And we did some quality control to remove those sequences that were just kind of yucky. Then, because we had taken a tissue that's both plant and bacterial, we took the genome of chickpea and removed all those sequences that match the chickpea genome, so that we're only left with DNA sequences that come from the bacteria. And out of this, we tried to make sense of that sequence. So we took 31 genes that are conserved across bacteria and used those sequences to build a phylogeny and to estimate the composition of bacterial species in those nodules. So what I've done here is sorted some of these nodules based on what bacteria is in them. So in many of these nodules, those that are predominantly blue, most of the bacterial sequences in these 31 genes belong to Rhizobia, some form of Rhizobia or another. But these nodules have lots of other bacteria in them, and even those that are mostly Rhizobia have some contamination from other bacteria. This is because we only washed off the surface of the root. So usually there's mostly Rhizobia inside, but there are other bacteria in the soil. The soil is incredibly bacterially rich, and we capture those. And some of these nodules were starting to senesce, and in them, in senescence, the Rhizobia are going dormant or lost entirely, and other bacteria, sort of soil saprophytes or other types of bacteria, end up invading the nodules. So our analysis shows this rich diversity in different types of bacteria in these nodules. We can ask a variety of questions. How does the genome content of the Rhizobia or the other bacteria vary? And do different chickpea species, the cultivated chickpea or the wild species, choose different symbionts? So we can assemble all these bacterial sequences and try to answer some of these. So we see differences in species abundance across these across the nodules that we sequenced from. And if we just take the blue ones, the Mesorhizobium, they fall into two groups of Rhizobia, both Mesorhizobia. One group, based on a subset of genes, belong to Mesorhizobium mediterraneum. This is a Rhizobia that has been previously found to occur with chickpea in Spain and in Turkey. The second group is a different Mesorhizobium. It appears to be a Mesorhizobium pluriferium that has been found to occur on acacia in India. And we find this across a couple different genes. So it looks like, although not shown well here, if I go back, that most of these group 1 Mesorhizobium were found on wild chickpea that we grew in India, while most of these group 2 occur on cultivated chickpea when grown in India. So we see this across several different genes, that there are these two groups. And part of what this suggests is that cultivated chickpea in India is occurring with different sets of Mesorhizobium. So I wanted to talk with you about this to give you a flavor of some of the things we can do. So here I'd like to thank many of my partners, including collaborators at UC Davis and in India, and our funding from the Gates Program, the National Science Foundation, and USDA. Thank you.